Bismillahirrahmanirrahim. Dear students, Assalamu alaikum and welcome to office.com.pk. Dear students, we are discussing about the cell cycle, and previously we discussed about the interphase, which is the preparation of a cell for cell division. Now today we are going to discuss the mitosis, which is actually the apparent cell division. Now, what is mitosis? It is basically the type of cell division which ensures the same number of chromosomes in the daughter cell as in the parent cell, which means that parent cell and daughter cells will have same number of chromosomes. So there is a slight difference between the cell division or the mitosis of plants and animals, but for convenience we will be discussing the mitosis of animals. So in the end I will also tell you what are those simple differences between the cell division of animal cell and the cell division of plant cell. Now the basic importance of mitosis is that it ensures the growth, okay, and it can also take place in haploid cells as well as in diploid cells, and it occurs in all parts of the body whenever it is required. So it is a continuous process, and it is divided into two phases. These are the subphases. The first one is karyokinesis, and the other one is cytokinesis. Now the karyokinesis involves the division of nucleus, means the whole nuclear division will be known as karyokinesis because the word karyo stands for nucleus. And the cytokinesis, basically it accounts for cytoplasm. It occurs after karyokinesis. Basically it divides the whole cell. So first of all, let us discuss karyokinesis. Now karyokinesis is, as you all know, is the division of nucleus. So when the process starts in animal cell, it starts with the partition of centrioles. You all know that centrioles are present at centromere, that specific point in the animal cell, and this whole centrioles or the centriole apparatus is duplicated during interphase, and it is present in the same centromere, means one centriole is divided into two centrioles, and the copies are also present at the same site. So early in mitosis, the two pair of centrioles separates and they migrate to the opposite side of the nucleus, and this will establish the bipolarity of the cell. Now let me explain with the help of a diagram. This is a normal animal cell. When it is preparing for these are the chromosomes, this is nuclear membrane, and here basically two centrioles are present. So when they are duplicated, they become four, and then they move to the opposite poles. The two pairs, they move to the opposite poles, and what happens? Something like this is obtained. Again, the chromosomes. Now the point where centrioles are present is known as centrosome, and the actual organelle is centriole. So two pairs will move to the opposite side, and this will create a bipolarity of the dividing cell. Now what happens next is that now I will draw a magnified diagram. Now this is the cell. This is for the idea of nuclear membrane. Here we have two pairs of centrioles present at opposite poles of the cell. Now three types of fibers or microtubules, they start originating from the centrioles. There are three types of these microtubules. There are some microtubules which are not related to handling of chromosomes. So these are known as astral microtubules. Because they do not contain the kinetochore connecting proteins, so they will form the ester. Okay. Now the microtubules which will run from pole to pole, one pole to the next pole, they will connect from pole to pole, they will be known as the polar microtubules. And the ones which will handle the chromosomes are known as kinetochore microtubules. Because they will attach at the kinetochore of the chromosomes. So three sets of microtubules, they will originate from each pair of centriole. The first one are the astral microtubules, which are not involved in the handling of chromosomes, and they will be known as astral microtubules. The other two sets, they are involved, the polar microtubules and the kinetochore, they will form the spindle. Okay, so the polar microtubule, they will run from one pole to another, they will interact with the opposite poles, and the kinetochore microtubules will be handling the chromosomes. Now what will happen next, that the microtubules, their composition, now they are composed of protein which is known as tubulin, and there are few traces of RNA which are also present. Now this specialized microtubule structure, which will include ester. Ester are the fine microtubules that are not involved in the handling of chromosomes, plus the spindle. The ester and the spindle together is known as mitotic apparatus. The ester plus spindle. Now this mitotic apparatus will be larger than the nucleus. As you can see, it is covering almost whole area of the cell, and it is designed specially to attach and capture the chromosomes. The mitotic apparatus will attach, capture, align and separate the chromosomes so that the equal distribution is ensured, because we know that mitosis is the cell division which will ensure the equal division of the chromosome in the daughter cells. So we have to make sure that the chromosomes are equally divided. So this function is performed by this mitotic apparatus, which is composed of ester and spindle, and it will attach with the chromosome, then they will capture them, then they will align them accordingly, and finally they will separate them equally, making sure that the equal distribution is ensured. Now karyokinesis is further divided into prophase, metaphase, anaphase, and telophase. Now let us discuss all these phases one by one.